Hello, my name is Joshua Mugabe. I welcome you to Mendel's Laws. Who is Gregor Mendel? Mendel is considered the father of genetics. He was a monk, biologist, and a botanist born in Australia in 1822 and who died in 1884. During the year of 1853 and 1863, he cultivated peace plants in the garden in his monastery to be used for his research. His experiments consisted of crossing pea plants of distinct characteristics, that is size, color of the seeds and so on, categorizing the results and interpreting them. The experiments led him to enunciate his laws, published in 1886 with no specific scientific repercussions at that time. Only in the beginning of the 20th century, in 1902, 18 years after his death, were his merits broadly recognized. What in genetics is hybridization? Hybridization in genetics is the crossing of individuals from pure and different lineage in relation to a given trait, that is, the crossing of different homozygous for the studied trait. In Mendel's experiment with peas, for example, plant from a green pea lineage obtained from self-fecundation of its ascendant through several generations was cross-fecundation with another plant from the yellow lineage, also obtained by self-fecundation of the ascendant. The self-fecundation through several generations of ascendant and exclusively obtained of individuals with the desired characteristic assured that individuals of parental generation were pure, i.e. homozygous for the characteristics. What is monohybridizing? Monohybridism is the study of only one characteristic in the crossing of two pure individuals, that is hybridization for the characteristic. Considering hybridization in traits like color of the flower of a given plant species, red dominant, yellow recessive, conditioned by a pair of different alleles, what are the phenotypic results of the first generation F1, the phenotypical result of the second generation F2, formed by crossing among F1 genotypes? What are the phenotypic proportion of F1, F2? In relationship to genotypes and phenotypes, the hybridization comprises of parentogenelation, P, red, RR, yellow, YY. The F1 generation will be RR multiplied by YY, and we get RY, red. The F2 generation, RY and RY, we get R, capital R, capital R, capital RY, capital RY, and YY. In the F1 generation, the proportion of the red flower is 100%. In the F2 generation, the phenotypic proportion of the red flower is 75 and yellow is 25. Hybridization in a trait, like color of the flower of a given plant species, red stroke yellow, conditioned by different alleles in relation to complete dominancy, red dominant, yellow recessive. Why in F1 generation is only one color missing? In this monohybridism, one of the color does not appear in F1 generation because their parental generators are pure, that is homozygous. And in F1, all descendants are heterozygous. Each parental individual forms only one type of gamete. Since only heterozygous genotype appear, the red is dominant over yellow. The individual of F1 generation will present only red flowers. Considering hybridization in the gene traits like color of hair of a mammalian species, white and black, conditioned by different gene alleles under complete dominance. Black dominant, B recessive, white. How can the phenotypical proportion obtained in F2 generation be explained? What is this proportion? In the monohybridism, conditioned by two different alleles, the F1 generation present only heterozygous individual, B, W. In F2 is one individual, capital B, two individual, capital B, small w, and one individual, small w, small w. In relation to the phenotype, there are in F2 black individual, one white individual. Since black is dominant color, so the proportion is 3 to 1, 3 black-haired to 1 white-haired. What is meant by saying that the relationship to a given trait conditioned by a gene of two different alleles, the gametes are always pure? To say that gametes are pure means that they always carry only one allele of the referred trait. Gametes are always pure because all in them the chromosomes are not homologous. They contain only one chromosome of each type. What is Mendel's first law? Mendel's first law postulates that a characteristic trait of an individual is always determined by two factors, inherited from the father and another from the mother, and the direct offspring of the individual receive from it only one of these factors. In other words, each trait is determined by two factors that segregate during gamete formation. Mendel's first law, is also known as the law of purity of gametes. Mendel deduced the way genes and alleles are transmitted and traits were conditioned without even knowing the existence of these elements. Which is the type of gamete for a given trait produced by a dominant homozygous individual? What are the genotypical proportion of these gametes? What about a recessive homozygous individual? If an individual is dominant homozygous, for example, capital A, it will produce only gametes having the allele capital A. A proportion that's 100% of capital A gametes. If an individual is recessive homozygous, for example, small a, it will produce only gametes having allele A, also 100% proportion. Which is the type of gamete produced by heterozygous individual? What is the genotypical proportion of these gametes? Heterozygous individuals, for example, capital A, produce two different types of gametes. One containing allele A, another one containing allele small a. Proportion is 1:1. If F2 generation of hybridization for a given trait condition by pair of allele capital T, small t, according to Mendel's first law, what are the genotype of each phenotypic form? How many respective are the genotypical and phenotypical forms? In the mentioned hybridization, the genotypical form in F2 will be capital T, small t, capital T, small t. Therefore, there will be three types of genotype forms and two different phenotypic forms, that is capital T and small t. Capital T dominant over small t. Why can the crossing of an individual that manifests dominant phenotype with another that manifests recessive phenotype for the same trait determine whether the dominant individual is homozygous or heterozygous. For crossing the individual having recessive phenotype with another individual having dominant phenotype for the same trait, it is possible to determine whether the dominant individual is homozygous or heterozygous. This is true because the genotype of the recessive individual is obligatorily homozygous. For example, small a. If another individual also homozygous, capital A, the F1 offspring will be heterozygous. As capital A small a is equal to only capital A small a. If another individual is heterozygous, there will be two different genotypical forms, capital A small a, in the ratio of 1-to-1 proportion. So if the recessive phenotype appears in direct offspring, the parental individual can manifest dominant phenotype of certain heterozygous. What is the genetic family tree? Genetic family tree is a semantic family tree that shows biological inheritance of some trait through successful generations. Genetic family trees are useful because it's typically impossible and ethically unacceptable to make experimental crossing for genetic testing between human beings. Within the help of the family tree, the study of analysis of marriage and crossing that have already occurred in the past. From the analysis of the family tree, for example, information on probability of the emergence of some phenotype and genotype, including genetic diseases in the offspring of the couple, can be obtained. What are the main conventional symbols, signs used in genetic family trees? In genetic family trees, the male sex is usually represented by a square and the female by a circle. Crossing are indicated by horizontal lines that connect the square to circle, and their direct offsprings listed below are connected to the line. The presence of a studied phenotypical form is indicated by a complete Hatcher shading of the circle. The square corresponds to the affected individual. It is useful to enumerate the individual from the left to right or from top to bottom for easy reference. What are the three main steps for the good study of a genetic family tree? Step 1. Determine whether the studied phenotypical form has a dominant or recessive pattern. Step 2. Identify the recessive homozygous individual. Step 3. Identify the remaining gametes or genotype. What is Mendel's second law? Mendel's second law postulates that two or more different traits are also conditioned by two or more pairs of different factors that each inherited pair separates independently from the other. In other words, gametes are formed always within an oriented representative of each of the factors that determine the phenotypical characteristics. Mendel's second law is also known as the independent segregation factor or the law of independent assortment. What is the condition of Mendel's second law? To be varied. Mendel's second law is only varied for genes located on different chromosomes. For genes situated on the same chromosome, like linked genes, the law is not varied, since segregation of this gene is not independent. According to Mendel's second law, in the crossing between homozygous individuals concerning two pairs of non-linked alleles, capital A, capital A, capital B, crossed with small a, capital B, what are the genotypical and phenotypical proportion in F1 and F2? The parental genotype, capital A, capital B, capital B, capital B, small a, capital B. Gametes from the parental generation, capital A, small b, small a, small b. Thus, F1 will have 100% capital A, capital B, small b gametes, and a phenotypical correspondent form. As F1 a capital A, capital B, small b individual, the gametes from crossing can be capital A, capital B, capital A, small b, small a, small b, small a, small b. The causal combination of these gametes forms the following genotypical forms. 1. Capital A, capital B, 2. Capital A, capital B, small b, 2. Capital A, capital B, small b, 4. Capital A, capital B, 1. Capital A, small b, small b, 1. Capital A, small b, small b, 1 small a, capital B, 2 small a, capital B, small b, and 2 small a, capital small b. the phenotypical proportion then would be 9 capital A dash, capital B dash, double dominant. 3 capital A dash, smaller b, dominant for the first pair, recessive for the second. 3 small a, capital B dash, recessive for the first pair, dominant for the second. 1 small a, smaller b, double recessive. Considering independent segregation of all factors, how many types of gametes does capital V, small v, capital X, capital W, small w, capital Y, small z, individual produces? What is the formula for determining such number? The mentioned individual will produce 8 different types of gametes. Attention, gametes and not zygote. To determine the number of different gametes produced by a given multiple genotype, the number of heterozygous pair are counted. In the mentioned cases, 3. And the result is placed as an exponent of 2. For example, 2 to the power 3, which is equal to 8. How is it possible to obtain the probability of emergence of a given genotype formed of more than one pair of different alleles with different segregation from the knowledge of the parental genotype? Taking as example of crossing capital A small a, capital B small b, capital C small b with small a, capital B, capital C small c. For each considered pair of allele, it is possible to verify which genotype can form as an independent analysis and in the proportion. Capital A, small a, capital A, small a, small a in a ratio to 1 to 1. Capital B, small b, crossed with capital B, capital B, smaller b in a ratio of 1 to 1. Capital C, small c, capital C, small c, crossed, capital C, small c, small c in a ratio of 1 to 2 to 1. The genotype to which the probability is to be determined is, for example, smaller a, capital B, small c. For each type of this genotype, the formation probability is determined. 2 smaller a 0.5, 2 capital B, 0.5, 2 small c 0.25. The final result is obtained by multiplication of this partial probability, 0.5 times 0.5 times 0.5, resulting in 0.06235. Thank you very much for participating in Mendel's Law. Please do like, share, and subscribe.